3.
Hello, welcome everyone. Good morning and good evening. Thank you for coming to this event, the second transcontinental seminar in our joint TWRA RMIT webinar series focusing on biodiversity assessment. I am Monica Tuman from the Aquatic Research Group at RMIT University in Melbourne, and I am your facilitator today. I'm coming to you from the lands of the Wurundjeri people, on whose unceded land we conduct the business of the university. We respectfully acknowledge their ancestors and elders past and present. We acknowledge the traditional custodians and ancestors of the lands and waters across Australia and Brazil, where we conduct our business. This joint partnership is between, from Brazil, the Tropical Water Research Alliance, TWRA, and from Australia at RMIT University, the Aquatic Environmental Stress Research Group, and the Water Effective Tools and Technology, or WET group. We have the aim of increasing our combined research impact by facilitating opportunities and knowledge sharing, which leads us to events like today. We will hear from 4 experts about their research in biodiversity assessment, 2 from RMIT, Professor Jeff Shimida and Dr. Sarah Long, and 2 from TWRA, Drs. Yara Moreto and Paula Oliveira. Following this general introduction, I'll introduce researchers individually, who will speak for about 10 minutes each. I've got a little bell I'll ring if we go to 12 minutes, just to let people know that they need to wind up. And at the conclusion of the 4th presentation, we'll have a bit of a panel discussion with all the speakers. So during the presentation, audience, if you have any questions, please type them into the chat and I'll pose them to our panel. We are also recording this as well as it being live streamed. And just to reiterate, please make sure you have your microphones on mute if you're in the audience, and your video off unless you are presenting or on the panel. So first up, we have Jeff Shimida, Professor Jeff Shimida, who's actually affiliated with both of the research groups at RMIT University. Jeff is a marine ecologist working in several areas of aquatic pollution and conservation related to freshwater as well as marine environments. He comes from a background in biological oceanography with positions in the US at the Woods Hole Oceanographic Institution, UC Berkeley, and Franklin and Marshall College, followed by Melbourne University before settling at RMIT in 2008. His recent main research areas have been in invertebrate larval settling processes, development of biofouling communities and design of novel anti-fouling technologies, impacts of herbicide use in salt marsh ecology, eDNA studies of dinoflagellate plankton in Australian waters, and impacts of microplastics on aquatic invertebrates. He supervises PhD students using eDNA approaches to investigate impacts of pollution on microbial communities. And with that, I'd like to invite Professor Jeff to start sharing his presentation and talk.
Well, thank you very much, Monica. I appreciate the introduction and especially the invitation to speak here to everyone. I'd like to say good day to everyone at the TWRA, our colleagues in Brazil. It's nice to be here with you today and share a bit of the work that we've been doing. So I'll share my screen. Here we go. I think this is going to work. Do you see that now? Okay, great. So the research I'm going to talk about today is on using eDNA metabarcoding to evaluate biodiversity and its drivers in marine biofouling communities in a couple of different contexts. I'd like to thank my collaborators in this research, Martin Leary and Nathan Bott from RMIT, Also Richard Piola and Andrew Scardino from the Australian Defence Science Technology Group. And this work involves students, Sam Tyrrell, an honors student, Matt Watson, a PhD student, Gemma, an honors student, Megan, a PhD student, and Renee, a master's student. By the way, can you see my cursor as I point to things?
Yes, we can.
Great. And there we go. All right, so for those who might not be familiar, marine biofouling refers to the growth of organisms on materials in the ocean that is unwanted, so something causing nuisance. I've got a few photos here. Down in the bottom right is a ship hull. You can see a lot of growth on it. This in the lower left is an autonomous vehicle, a glider that measures seawater properties remotely, and this is a casing around an instrument deployed in the ocean, and you can see a lot of growth on them. There are barnacles and mussels and tube worms on these. That's referred to as hard fouling, but there's also soft fouling, which you can see in this picture here, that includes things like sponges and sea squirts, hydroid colonies, and macroalgae. So this this phenomenon afflicts a lot of different types of surfaces in the ocean on materials that are of interest to various industries. So I've got listed here some places you find a lot of biofouling: on ships, in aquaculture, on instrumentation used by scientists and others, and a variety of types of infrastructure. There we go. This biofouling grows on really any surface that's submerged in the ocean. It starts within seconds, as shown in this graph here, with the absorption of organic molecules onto surfaces. And this is what's called a conditioning film that develops. It also includes the initial attachment of bacteria. From that, it develops into what we call a biofilm, which includes not only microbes growing, but also their exudates, extracellular polymers that create a matrix. And following that, you get colonization of additional microbes, including eukaryotes, and algae, and larger organisms, or macrofouling as it's called, which are the animals and large algae. Now, they don't necessarily follow this successional sequence. What's certainly typical is the substrate conditioning followed by the microbial settlement. So that's the microfouling stage. The macrofouling stage can happen in various successional stages, depending on, say, availability of recruits at the time. So it's not necessarily such a predictable sequence, but we can distinguish between the conditioning film, the microfouling, and the macrofouling, which can be both soft and hard organisms, as I said earlier. Now, I'm going to emphasize microfouling in this talk for a couple of reasons. First of all, the microfouling itself is problematic, even though that's not the most notable or noticeable to the eye. Like in the photos I showed earlier, that was all macrofouling you were looking at. But the microfouling, that microbial scum in the biofilm itself, is problematic because it contributes to corrosion of materials, and it creates drag forces. It can reduce the flow of water across surfaces and affect the performance of things like ship hulls and various instrumentation in the ocean. Also, the microfouling acts as a cue for the settlement of some macrofoulers. It's well known that the presence of microfouling organisms or their chemicals from them can be cues that stimulate the settlement or inhibit settlement of macrofoulers. And so the actual assemblage structure of the microfouling has implications for the nature of the macrofouling that develops. So we've been researching questions such as these. To look at the microfouling assemblages, we've been documenting the natural variation using eDNA technology and asking what sort of natural variation exists in different habitats, and especially associated with aspects of water quality that might be driving the particular assemblages that develop. Secondly, in what ways do those microbial assemblages influence the subsequent macrofouling, as I said, because they can serve as cues for the settlement of larger organisms. And as Monica mentioned, we've been working on some antifouling treatments. There are common antifouling paints like copper paints that are put on surfaces to restrict fouling, but they still allow microfouling assemblages to develop. So we want to examine what type of assemblages are still there. And as I said, we've been using eDNA metabarcoding to do this. All right. Boy, I see time is passing quickly, so I'll try to speed up a bit. Here's a study looking at the natural variation of microfouling assemblages where we made measurements at 4 different sites. 2 were urban sites around Australia. Here's a map of Australia. We're in the state of Victoria and down by Port Phillip Bay. This is Western Port Bay next to us. And we had 2 sites that were urban in Hobson's Bay and Warneet, 2 sites that were coastal. And so you would expect the water quality to be quite different between these. We deployed these surfaces, which were Petri dishes, in the water for up to about 6 weeks to monitor the development of the microfouling. And the advantages of these little plates are that we can cap them before we take them out of the water. And so they enclose a little bit of the overlying water. So we can look not only at what's embedded in the biofilm, but also what's loosely associated with it and what's in the surrounding water. And we did this by extracting and sequencing the 16S RNA. Here is a bubble plot of the diversity of microbes that we found at these sites. So Blairgowrie and Flinders are the 2 coastal sites. Hobson's Bay and Warneet are the 2 urban sites. In each case, we've got measurements at 2 weeks, 4 weeks, and 6 weeks. And here on the right are samples from the immediately surrounding water at each of those locations. So here are some taxa of bacteria, and the size of the bubble indicates their relative abundance. And you can see there are quite a lot of differences among the different sites we sampled, and they differ quite a bit from the water. So a few conclusions from this: the biofilm assemblages are very distinct from the surrounding water. So it's really a unique community that is specialized for growing in these biofilms. And there are some clear differences in taxa among these different sites. And of course, compared to the water samples, I'll just point out some that may have implications for the further development of biofouling, like the Flavobacteriales, which are shown in this row right here. You can see there are more of them in Hobson's Bay and Warneet, the 2 urban sites, than in the coastal sites. And these are of interest because these are known to serve as cues that enhance the settlement of macrofouling animals and algae that are quite problematic. Furthermore, the overall diversity, the biodiversity, was lower at the 2 sites that are more urbanized. And this is a plot showing how the assemblages from the 4 sites shown in the symbols differed from each other in a principal coordinates plot. We've got the 2 urban sites here, the furthest left, and the 2 coastal sites more to the middle and right. So they are distinct from each other in their assemblage structure, and they separate according to water quality parameters. So the 2 more urban sites are associated with lower dissolved oxygen, higher phosphorus and nitrogen content, higher dissolved solids, and less salinity and lower pH. So clearly there was separation of assemblages that was related to water quality parameters. And so the conclusion here is that the water quality itself may be one of the natural drivers that influences the assemblage of microfoulers that we get. And this can also indirectly affect macrofouling structure because, as I said, certain bacteria are known as settlement cues. We've also done some research and published it on the role of protozoans as settlement cues for macrofoulers. Some of them can facilitate settlement, some can inhibit settlement. All right, and then the second thing I want to mention briefly is some of our work on how microfouling assemblages respond to antifouling treatments. As I said, there are common paints like containing copper or other poisons, and also coatings that are hydrophobic, like silicon-based coatings that promote the release of fouling organisms. It's essentially, it's hard for them to hold on. And as a ship, say, starts passing through the water and there's some water movement, these organisms will easily be detached. So we know that these types of coatings are very effective against macrofouling, but they still develop microfouling coatings. There is still a slime of biofilm that develops on these, which, as I said, can be problematic in terms of corrosion and drag forces on the surface. And we know that microfouling can influence subsequent macrofouling. So even if these biocidal paints like copper are effective for a while against macrofoulers, they still can develop, and that may be influenced by how the biocide is altering the microfouling community. So we did a study with a variety of coatings to see how they influence the microfouling community. Now I'm showing you some plates that we deployed in the ocean for up to 17 weeks. These are photographs of controls where you can see a lot of tube worms, tunicates, bryozoans, etc. On the bottom are plates coated with 3 commercial antifouling coatings. This one has a copper biocide. This one here is a silicone-based fouling release, and this is fouling release with some copper. And you can see there's much less growth on there. It's mostly some algae and detritus and a biofilm. There's clearly a biofilm on here. Part of this study was also to see if we could produce 3D printed materials with plastic polymers, PCl and PLA, that had biocides embedded in them. So in one, we embedded copper. In another, we embedded a toxin called decoit. And you can see that these didn't have any macrofouling, but they did have some algal growth and detritus settling on them. The graph here shows that the macrofouling community was quite diverse on the 2 controls, the PLA and the PCl, corresponding to what you see in the photo. And the other treatments had much less diversity, but some of them had quite a bit of, say, in this example, microalgae in the light green covering the decoit. That's what you see here in this colored bits. The others were covered mostly by detritus or bits of microalgae, but they differed quite a bit. So clearly the biofilms do differ among these biocidal treatments. And we looked further then at the composition of the microbes by, again, doing 16S DNA sequencing. This is a graph showing bacterial or prokaryotic classes. And we also did 18S sequencing for eukaryotes. So I've got some information on eukaryotes down in the conclusions here. But I'll just point out that there are clearly some very large differences in the prokaryotic assemblage structure. Here's the decoit compared to our control, and you can see that increase of the microalgae due to the chloroplasts here, and we identified those as green algae. You can see some differences in the flavobacteria, in the beta protobacteria, and I've listed the main take-home conclusions here, so I'll just go through those. Our decoit biocide clearly reduced the biodiversity overall. It inhibited cyanobacteria and diatoms and the flavobacteria. And this is really important because those flavobacteria, as I said before, they're positive cues for hard foulers. So the decoit not only can inhibit the macrofoulers, but it can inhibit the bacteria that favor them. But it did stimulate a green algae microfouling film. The copper biocides, that's the copper 3D printing we did, and also the copper coatings that we put on, they reduced microalgae across the board, including cyanobacteria, diatoms, and chlorophytes. Thanks for the bell, Monica. I'm almost done. They reduced gamma protobacteria, at least on our 3D printed one. And that's interesting because some gamma proteobacteria have very strong impacts on macrofoullers, either positive or negative. So we'd have to drill down further to look at the species structure to infer what the implications could be. But they favored the flavobacteria. So that does the opposite from the decoy. It provides more positive cues for hard foulers. So if the copper isn't effective for very long, it could be that the influence on the microbial assemblage could actually stimulate more development of hard foulers. And the fouling and release coatings, they stimulated the beta proteobacteria, and there's very little known about how they could influence macrofouling. So to conclude, we've found a lot of new insights into microbial assemblage variation using eDNA metabarcoding, both in terms of how it relates to natural water quality parameters and also to the anti-fouling agents that are applied. What I'd really like to pursue further is to see how these direct effects on the microfoulers could have follow-on indirect effects for the macrofoulers. And as Monica mentioned in the introduction, we're also working on some anti-fouling technologies, which would be the topic of another talk, such as altering the microtexture of surfaces to inhibit settlement and combining that with surface chemistry modification. So thanks very much. I'm sorry if I went over time.
Thank you, Jeff. That was fantastic. Very interesting work on biofouling. I'd now like to introduce Dr. Sarah Long and invite her to speak. Also, just a reminder that if you have any questions or you'd like to discuss anything about Jeff's talk, if you can put your notes into the chat. So, Dr. Sarah Long, after completing her PhD at RMIT on the effects of crude oil on marine invertebrates, Sarah has worked as an ecotoxicologist at the Centre for Ecology and Hydrology, CEH, at Monkswood in the UK. Here, she investigated the effects of exposure patterns, acute, chronic, and intermittent, of pesticides and industrial contaminants on detoxification responses in terrestrial animals. She was also involved in a European Union-funded Framework 6 project assessing the risks of chemicals on biodiversity. She managed a collaborative project between CEH and the University of Cambridge, developing small metabolite markers of pesticide exposure using novel metabolic techniques. And on returning to Australia in 2009, Sarah worked originally at the University of Melbourne with the CAPM Research Group, which is what we have morphed into at RMIT as the AQuest Group. Her main research focus has been developing methods to measure and understand biological effects of environmental stressors in local Australian species that can be used in biomonitoring programs to assess the condition of waterways. She's interested in understanding how environmental stressors, as well as natural ones, are toxic to organisms, using a range of techniques to better understand the mechanisms of toxicity, such as metabolomics and enzyme assays, as well as measures of fitness, such as growth, reproduction, and fecundity, and looking at early warning biomarker responses. With that, I would like to invite Sarah to begin her talk. Thank you.
Thanks, Mon. Thanks, everybody. Can you see this? Can you see my screen?
Yes.
Okay, great. Yep, thank you. Thanks so much for the introduction, Monica, and thanks everybody for coming to this talk. So I'm going to talk to you today about some of the work that we've done, basically the process that we use for unearthing contaminant risks to manage biodiversity at our really important sites. And this is a work that we do as part of the A3P, so the Aquatic Pollution Prevention Partnership, which is a collaborative partnership between Melbourne Water and RMIT University. So I'm just going to talk you through the process or the framework that we use for assessing contaminant risk to understand impacts of contamination on our sites of high biodiversity significance. So the first thing we do is we carry out a desktop review. So we want to assess the potential risk. And the questions that we mainly focus on at this stage is, what are the key values? So For us, the key values are the environmental values, which are things like birds, platypus, fish, frogs, macroinvertebrates, and vegetation. So we want to know what values are present at the site. And we also want to get an understanding of what the land uses are surrounding the site, because we know there's a link between what the land use is and also what potential contaminants are present at that site. So this is the criteria that we use for our risk assessment, for our matrix. So like I said, we look at what environmental values are present at each site. And we do tend to focus on the ones that we think are going to be more impacted with pollution. We also look to see if there's any previous reports, if they've identified pollution present. So things like stormwater, nutrients, and maybe herbicides based on the vegetation management around the site. And like I said, we also look at land use. So we know that's really important, the land use surrounding the site, because you know there's different contaminants at in different land uses. So we try and produce maps like this. Can you see my cursor? I assume that you can. So what we do is we create, oh, thanks, Mon. We create a map, and so each of the colours represents a different type of land use. So this is our site of interest. It's in blue. And so what we look at is what the land uses are in the catchment and further up the catchment. So you can see this area here is green, so that's grassland. So we know the types of contaminants that are likely to be used for grassland management. Anything that's red is industrial areas. And again, we know the types of contaminants that are present there. And then the purple is residential areas. So by looking at this map, we can get an idea of the types of contaminants that potentially are present at the site and maybe flowing into the site or coming onto the site through drift or through stormwater. So then what we look at is the impact of these contaminants on the value. So we look at a range of different databases based on our understanding of the land uses and the chemicals and the values that are there. So one of the main databases that we use is the US EPA Ecotox database. It has a lot of toxicity information on a range of contaminants and on a range of different species. So that's one of our go-to databases where we get some information, and that helps us to understand what the risks are to each of the values based on what we think, what chemicals might be there. So then we hope to answer this question: is the risk high? Is there a high risk of contamination to our biodiversity at this site? If the answer is no, which is great, we'll then just let the waterway manager know, and maybe would suggest just keeping an eye on what's going on at that site, just to make sure that the risk is still low. If the risk is high, then we obviously need to go and get some information. We need to validate what we think is going to be there based on our risk assessment. So we do some site assessments. So this is the workflow that we use for our site assessments. So first of all, we always do a site visit. We look to see if there's really where is a good areas to collect some samples. So we'll go and visit the site, hopefully get some information from local waterway managers, just based on their experience. Look at the surrounding areas. Are there any drains that we need to sample as well as the waterway? Then we take some samples. So this is a shovel that is full of sediment. So we just take the top couple of centimetres of sediment that shows the most recent deposition. We then filter the sediment through nets. So we only want to look at the fine sediment because that's where a lot of the contamination is absorbed. So that gets sent off. We mainly focus on, in the initial screening, metals, total petroleum hydrocarbons, and a range of pesticides. So we screen for over 100 pesticides, ranging from insecticides, fungicides, and herbicides. We also take some water samples, which we can see here. So again, we look at general water quality parameters, but we also measure metals and hydrocarbons within the waterway as well. And finally, we deploy passive samplers, as you can see in this picture here. So these are membranes that we use that we find are really effective in understanding different types of pesticides and different types of chemicals. So we have 3 different passive samplers that we use. that can detect chemicals ranging from water-soluble chemicals through to hydrophobic chemicals. And these include pesticides such as organochlorines, organophosphates, and some synthetic pyrethroids, through to pharmaceuticals and some personal care products. And you can see in this picture here, we deploy the passive samplers in the cage. They're submerged for up to 4 weeks. So we can basically get an understanding of what chemicals might be there, because not everything goes into the sediment. And then we retrieve all of the samples and they get sent off for analysis. So then we want to ask the question, are contaminants an issue? So hopefully the data that we get from our site assessments can help us answer that question. If they're not an issue, and so what we mean by that is we look at a range of different guidelines. So for contamination, we look at Australia and New Zealand marine and freshwater sediment and freshwater guidelines. So if the contaminants are below these guideline values, they're not likely to be an issue. We also look at some state objectives as well. So we're in Victoria. So the Victorian EPA has set some objectives of water quality. So if our contaminants are all within the objectives and below the guidelines, then we can say that we think that it's contamination is not a risk to biodiversity. Again, we'll inform the waterway manager, and again, maybe recommend we keep an eye on this site and maybe do some routine monitoring just to make sure that contaminants are not an issue. If they are an issue, then we go and do some more detailed site assessments. And we do a number of different site assessments. It depends on the question that we're asking, the site that we're looking at. But these can include expanding our initial sampling. So this could be site 1 where the red is. So what we want to know is where's the chemical coming from. So we might do some extended sampling up and down some waterways. We also want to know what the extent is of the contamination. So we'll continue to sample further down the catchment or the watershed to get an understanding of where the contamination is and what the magnitude is, hopefully to identify the source. We also carry out some ecotoxicological experiments. So we'll bring sediment and water from the site, bring it back to the lab at RMIT, and we have a range of different species that we maintain in the lab that we can then use to determine the toxicity of the sediment or the water. So we have insects, crustaceans and gastropods, and we also have some plants that we expose as well. So we know that these animals have not been exposed to anything previously. So any change in the biological response, we know it's due to whatever's in the sediment or the water. We also carry out some in situ caging. So this is similar to a passive sampler, but we use a biological organism instead. So we use things like crustaceans, we use shrimp, we use some amphipods, maybe snails, and also some plants. So we put them in cages and we deploy them at the site, and they're exposed for 7 to 10 days. We bring them back and we look at the change in their biology. We know that the only thing that's changing is in response to what's in the water. So we can then see what's going on in terms of the biology. And then we also might do some macroinvertebrate sampling. Again, we look at the presence and absence of particular types of macroinvertebrates to give us an indication of whether the water is the quality of the water. And so hopefully we can then answer this final question, which is, can the source be identified? If we can, we then recommend potential management actions to try and reduce the impact of the source on the biodiversity. And if we can't, we just keep going back and doing some different types of assessments until we can answer that question. So now I just want to finish by talking about a couple of PhD student projects where they're looking at the impacts of contaminants on biodiversity. So the first one is a student, Madara. She's supervised by Professor Vincent Pettigrove and Dr. Claudette Keller. And her project is to look at the impact of synthetic pyrethroids, which is an insecticide that we're finding routinely in Melbourne on aquatic fauna. So she's looking at the impact on zooplankton and as well as macroinvertebrates in wetlands. And the second student is Adele. Again, she's been supervised by Vin and Claude, and she wants to look at the relationship between platypus and pollution by looking at macroinvertebrates as well. So I just want to finish with a few thank yous. So thank you to all the AQuest staff and students for field and laboratory support and also assistance. To Melbourne Water, to the project staff for their advice and support. The work was funded through the Aquatic Pollution Prevention Partnership. Like I said, it's a collaboration between Melbourne Water and RMIT. And I'd like to thank you all very much for listening. And if you'd like any further information, please contact myself or Monica for some more information. Thank you.
Thank you, Sarah. Really wonderful work. And next on our schedule, we have Professor Yara. Professor Yara is a biologist. She completed her undergraduate degree at the State University of Maringe, also going on to do her master's degree and then doctorate in ecology of continental aquatic environments through the graduate program in ecology of continental aquatic environments, PEA, at the University of Maringe. She has experience in the ecology of aquatic ecosystems, working primarily on the following topics: on benthic macroinvertebrates, biomonitoring, water resources management, and environmental impact assessments. Associate Professor, second order of the Federal University of Parana, Palatina campus. I'm sorry if I pronounce things incorrectly, since 2011. And the Professor of the Graduate Program in Environmental Engineering and Technology and Aquaculture and Sustainable Development. Yara was head of the Department of Biodiversity in 2016 and 17, then becoming the Deputy Director at the Palatina campus, and then the Director again through until currently. She's also the Vice President of the Tropical Water Research Alliance and is a visiting professor at the Australian Rivers Institute from Griffith University in Brisbane. With that, I'd like to invite Professor Yara to share your screen and start your talk. Thank you.
Thank you, Monica. Are you seeing my screen?
Give it a moment. It's saying that you're sharing your screen, but hasn't put your slide up again.
Okay.
So maybe share the online version as you did earlier, or is that what you're doing?
Yes, I'm doing that. Let me try again.
Okay. Yeah.
And now? Yeah?
Yes.
Okay.
Thanks.
So thank you and good evening to the Brazilian colleagues and good morning to the Australian colleagues. It's a pleasure to be here talking to you. And nice to see you again, Monica and Sarah. So today I'm talking about a project that was developed within the TWRA team regarding the influence of land use on aquatic ecosystems within the program Araguaia Vivo. This project is inside a bigger project called the Water Brazilian Program for Watersheds Revitalizations and was supported by Itaú Bank. And we developed this project, Sustainable Development Biodiversity Conservation of the Tocantins Araguaia Hydrographic Basin, under the general coordination of the Professor José Francisco Gonçalves Jr. And we have 3 different activities that were developed between 2021 and 2023 regarding restoration, biodiversity and biomonitoring, and land use, where the team was coordinated by Professor Edivando, me, Professor Daiane, and the researchers Dr. Natalia Stefanini and Dr. Marcelo Schmitz. So considering the biodiversity assessment it's important to know that degradation and fragmentation has led to major biodiversity losses. So we need to understand how this works, and also that strategies that rehabilitate fragmented landscapes and also try to conserve species are welcome to happen, and we need to invest more to have this kind of projects being developed in all our watersheds. So actions that increase habitat connectivity and also the ecological functions of the ecosystems are very important to give tools for decision supporters to make their better decision, and also to point the areas that are to be priority protected, and also the correct management of these regions. So in this project, the influence of land use on the aquatic ecosystems, I will present some final results because it was a huge project where we could do some models to the terrestrial and also to the aquatic ecosystems, and including the land use that you are seeing here in this figure, where we have the comparison of the land use composition in the Tocantins-Araguaia Basin among the years of 1985 and the 2020, where we can see that there is a replacement of the natural forest and the savanna dominance with the grassland, and also an increase of the agriculture and pasture areas. So it's very clear in the map that we have change in the colors. And also we could, with the land use model to the year of 2050, simulated based on the business as usual. That means the scenario that simulates the maintenance of the standard pattern of use that we have currently. And it shows that we will have also the dominance of soybean crop and an increase of the land use intensity until this year of 2050. So regarding this land use, we also have established the occurrence map for 51 species that were selected between according to an expert's consultation to produce some maps that I'm going to show in the next slides. So we have to this watershed a preview list of 1,395 species from the red list of the IUCN, and with the occurrence of mammals, birds, reptiles, amphibians, and fishes. And after to apply a questionnaire where we have the answers of 13 experts that did this consultation, they pointed out 51 target species that are very important economically and ecologically to the Tocantins-Araguaia watershed. And we increased our list of species to 1,458 species occurrence, and that were database georeference and recorded to the base, as you can see in the map. So we could bring to this project a first product that was the environmental data bank with the occurrence of target species. I mean, these 51 species that were target. Also the environmental variable description and the georeferential database to all the watershed. We also can produce critical models where were used the target species plus the environmental variables and the georeferential data to simulate and produce the models, ecological niche models, where we have the current climate suitability, as you can see here in green, the watershed of the Tocantins-Araguaia. And we did the projections to future climatic suitability to 2041, to 2070, and also to 2081 and 2100. So these climatical models and projections were produced to terrestrials and fishes groups as well. I will talk a little bit more about the aquatic species. So we have here the table that were produced to the aquatic species. So a group of fishes where we have 9 species target as needed to be protected and to have their area being prioritized. And all these data, they were collected and used by some platforms like HydroSheets to have the hydrological variables, the World Clean to have the climatic variables, share the socioeconomic pathways to have the carbon emission values that were used to produce some scenarios regarding 3 different patterns that we can have, and also the refuge maps that were crossed with some data about hydroelectric power station data from the Agency of Energy from Brazil. And here we have these 9 species that are very important also economically and ecologically. And the occurrence of each of these species was then aligned with a specific model protocol, considering only the water bodies of 4th order according to Strahler. So here we have the map with the climate refuge that were produced. You can see the red lines where we have the 27 climatic refuges for these 9 species that were modeled in 3 distinct scenarios. I mean, the optimistic scenario, where we have the carbon emission regarding 4.5, the intermediate scenario, 7.5 of carbon emission, and the pessimistic scenario that is 8.5 of carbon emission. And this map was constructed to the Arapaima-gigas, that is a species of fish very important economically to the Tocantins-Araguaia watershed, that is called pirarucu. It's a very big species of fish. So it's important to highlight here that we have to these species in the climatic refuges some hydroelectrical powers that are in operation, and also some that are with their viability studies approved. Here we have also the prioritization models that were produced considering 6 ranging classes, where we have the top 5 prioritization, I mean the highest first value area where we need to prioritize, the top 5 and 15 with the second relevant area, the top 15 to 25, the third relevant area, 25 to 50 to the intermediate, 50 to 80 below areas for prioritization, and the 20 that are the lowest prioritization areas. So in the figure A, you can see all the watershed with this prioritization. And here in B, we have the prioritization model overlapped with the climate refuges for fishes in these 3 categories. So it's important to highlight here that the important regions for prioritization, the top 5 and the top 5 to 15 categories, largely coincident with the stretch of climate refuges in the 3 climate scenarios. So it's an important data that we also have. And here we have the same scenario showing more clearly the 2 different carbon emission and the overlay of the UHEs that are the hydroelectric power plants that produce a lot of energy. And we can see again that we have the prioritization areas with some UHEs power plants. And that means that it certainly can produce losses on biodiversity if it's not managed correctly. And here in this figure 6, we have the overlay of the location of the small hydroelectric power plants, also in operation and planned to be implemented. And you can see that to the 3 different scenarios, we also have a higher number of PCHs, the small power plants, than the UHEs in number of enterprises. And it is, again, also largely coincide with the stretches of the climate refuges that are produced to this important ecological and economic fish species. So, and considering the optimistic scenario and also the pessimistic scenario, we also need to have some actions regarding the conservation and also the use, the land use of these areas. So quickly, as outcomes and conclusions of this small part of this big project, we can see that the importance of studies on use, influence regarding the biodiversity assessment, also the scope of the human impacts currently and also in the future, that these kind of tools are very important to be implemented and expanded to all basins and all groups of species. I mean that we need much more studies regarding this situation. And we do need prioritization areas as refuges to these species, especially considering that we need to reduce the fragmentation and that we also have impacted the areas as in the same place as the areas of prioritization. So it's a huge project. And we also need investments on carbon market emission, the ISG, to reduce the carbon drastic scenario that if you don't do anything, it will happen. To invest in sustainable agriculture, like the bioeconomy and the valuation of the ecosystem services, and also to have better talks with public, to produce public policies that guarantees the protection of these areas, to avoiding fragmentation, to avoiding the damage of prioritization areas, and also the losses of biodiversity. So I would like to thank you very much to everyone that are here, and muito obrigada.
Thank you, Yara. Such a large body of work showing, you know, the magnitude of what's being faced in the program that you've got in place. It's pretty amazing. Finally, I know we're running short on time, but I'd like to introduce Paula Oliveira, who received a PhD in aquatic ecology from the University van Amsterdam in Netherlands in 2019, and she's currently a postdoctoral fellow at ESALQ for Aquatic Biodiversity, coordinator at TWRA, and co-president of the Latin American chapter of the Society of Freshwater Sciences. She is interested in freshwater ecology with an emphasis on ecology applied to ecosystem restoration. Paula's research covers limnology, ecological restoration of rivers, benthic invertebrates, decomposition, and ecosystem processes. She's returned from maternity leave about 12 months ago, and I would like to invite you to start sharing your slides and give your talk, Paula.
Thank you, Monica.
Welcome. Great.
Yeah.
So if you go into presenter mode when you're done.
Can you see my screen?
Yes.
Yes, thank you very much for your presentation. So good evening for the Brazilian side and good morning for the Australian side. I'm very happy to be here today and to present a little bit of this work that I've been coordinating together with other colleagues from this key topic from the TWRA. So My talk today is entitled Trends in Freshwater Biodiversity Knowledge in Brazil, a quantitative literature study using topic modeling.
Paula, before you continue, you're on presenter mode, but it's showing.
Okay.
I'm not sure if you can swap it. Otherwise, we'll continue as is.
No, I can.
Because we can see your next slide and you're talking about
yeah. Let me try to.
Maybe swap display or something. Yep. Yeah, that should be the one. No, okay, continue.
Yeah, okay. So I will just continue like this.
Yeah,
I think one. Can I do like this?
You could. Yep.
Yeah. So I think one important point here is to show that we have eight key teams from TWRA, and this work was done by this key team 3, conservation, planning, and biodiversity assessment. So we are over 30 participants from this key team. This include professors, postdoc, and PhD candidates from all over the country. So this is Latin America, this is Brazil, and all this green circle are states with participants in this key team. So as an introduction of this work, I would first show you a little bit of Brazil. So Brazil is a country with 6 different biomes, and these are pictures of streams from these different biomes that range from wet forests like Amazon forest and Atlantic forest. We also have the largest wetland area in the world, that's Pantanal. We have natural grassland areas in the south, the Pampa, but we also have dry biomes like savanna in this green area, and the driest area, that's Caatinga. So we have 6 biomes over an extensive area in South America. That's wide variability of environmental conditions and a large number of ecosystems and high biodiversity and endemism. Brazil is a biodiversity hotspot, and freshwater biodiversity studies are, however, heterogeneous between communities and regions. So if you see here this yellow part, that's the Atlantic forest biome. We have most of the studies here because most of the cities, big cities are here, and university as well. So we have lack of studies in other areas such as Caatinga and maybe Pampa as well. So there are knowledge gaps over time, but also over regions in the country. So our aim was to access trends and knowledge gaps in the literature on freshwater communities in Brazil. So we selected 6 groups: macroinvertebrates, fungi, fish, phytoplankton, zooplankton, and meiofauna. And we run a literature review. So to do so, we selected keywords with 3 different groups of keywords. One related with the group. So for example, macroinvertebrates was our group of interest. And also studies related to community. So we were very focused on studies on community or assemblage in Brazil. So we first identify keywords with specialists that we named naive keywords. We did a literature review on Scopus and Easy Web of Science with these naive keywords. And following, we complete this list of keywords using this package named Lead Search on R, where we could, by doing so, we could identify missing keywords. So then we complete our first keywords list. So here you can see an example, and everything that's in red is this missing keywords that we found with this lead search package. After that, we finalize our paper selection. We check for duplicates or other issues, and we run a topic modeling using a LDA model. So the LDA modeling was used to identify common topics reported in our data set, in our papers pool. So we identify sets of co-occurring words, and by doing so, we defined, the model defined topic words. So this is an example of how to define a topic. So the model gives you some top terms. Here we used the 15 top terms. And so the topic one has this list of 15 top terms. And by checking these top terms, we can give a name for this topic. So for example, the topic one, we gave the name land use changes. This LDA modeling also capture the heterogeneity of the research topics within scientific publications and produces a matrix of wave of each word within each topic. So with this matrix of wave, we can produce some graphs such as NMDS to analyze this topic similarity. We could also identify topic popularity by mean topic proportion. So we could identify hot topics that were growing in popularity and cold topics that were decreasing in popularity. Okay, so we have some results here. So here is just a timeline and scientific publications numbers among different groups. And what we see here is that first studies are from early 70s with zooplankton, the yellow one. From 2005 on, more or less here, there was a rapid increase in community studies. Fish and macroinvertebrates, so fish is the blue, the dark blue, and macroinvertebrates, the orange one, were the most studied groups. And fungi, the green one, were the least studied group. Here is really small now. But what we can see here is that different groups, the different columns here, has different numbers of topics. Fungi had less number of topics, while macroinvertebrate and fish had 16 more topics. We could also identify some specific topics per group. So this yellow, highlighted in yellow, are specific topics. So for example, for phytoplankton, we could find cyanobacteria bloom topic, while for macroinvertebrates, this EPT biotic index. So we had these specific topics per group. We also had some common topics. The only common for all groups were plant associated. So we always found some type of topic that was related with plants. And beta biodiversity and functional ecology were also common topics for phytoplankton, zooplankton, macroinvertebrates, and fish. Okay, so for fungi, we had 31 publications and 9 topics. And here you can see one topic similarity graph, that is this NMDS, where we can see the most frequent topic, that's this larger circle. So the larger circle is topic 6, that's macroinvertebrate relationship, was the most frequent topic, and it was also one of the hot topics. So this is this analysis of popularity, where we can see if this topic was growing in popularity or decreasing in popularity over time. And here in the dotted red square, there were the hot topics, and the blue one, the cold topics. For phytoplankton, we had much more publications, so 469, 14 topics. And here we can see that floodplain, the number 2, temporal and vertical abiotic variation, the number 5, and cyanobacteria blooms, the number 4, were the most frequent topic. Floodplain was related with beta biodiversity here, and cyanobacteria bloom with fish farm impact. And for phytoplankton, there was no hot nor cold topic. For zooplankton, we had 409 publications and 15 topics. Trophic ecology in reservoirs, the number 9, was the most frequent topic. And there was a group of topics related here that was functional diversity, spatio-temporal distribution, and testate amoeba. And there was no hot topic. For meiofauna, we found 122 publications and 13 topics. Oligochaete in pristine systems, floodplain diversity, and large-scale diversity were the most frequent topics. And large-scale diversity was related with leaf litter associated. There were 4 hot topics and 1 cold topic for meiofauna. For macroinvertebrates, we found 722 publications and 16 topics. Benthic ecology and EPT biotic and index were the most frequent topics. And we could see that reservoir ecology here was related with larvae morphology. There were 3 hot topics and 2 cold topics. Okay, so the last one was fish. We had almost 800 publications and 16 topics. We had dam, reproduction and functional ecology, and feeding ecology was the most frequent topic. And dam was related with reproduction, and plant associated with landed ecosystem, and fishery management with urban changes. There were 4 hot topics and 3 cold topics. So as a discussion, what we see is that fungi and meiofauna lack community studies, thus our analysis should be seen with caution. Most topics were community specific, and there was only 1 common topic that was plant associated. Most topics were on fundamental ecology, that shows that applied ecology topics were limited to bioassessment and fishery management. Land use changes and fishery was the topics most related with societal issues, and few studies used experimental approach at community levels. So as a few remarks, what we see with this study is that we mapped the biodiversity literature landscape for Brazil and identified understudied research topic and potentially important to freshwater conservation. But one of the results that I think it's most important here is this collaborative effort of TWRA Brazil that has strengthened the network of regional researchers on freshwater biodiversity. And here is a picture of our collaborators in Brazil that I'm very grateful to work with them, and the institutions and fundings that we had to work, and yeah, by doing so to also produce this study. So thank you very much.
Thank you so much, Paula. We've gone a little bit over time. Thank you. Another, really large volume of work and a great presentation to round out our seminar. We had intended to have a panel session now, but we are over time. So I think we'll wrap it up from here. I'd just like to say, a very big thank you to all of our experts for presenting, and to all the people who have attended. And we really look forward to our next seminar in the series. Thank you.
Thanks, everyone.
Thank you, Monica. Thanks, everyone.
Thank you.
Thank you.
Bye-bye.
Bye-bye. Nice to meet you. Bye.
See you. Nice to meet you.